making a new individual is expensive, right? And generally, parents are the ones who are paying the costs. So eggs are physiologically costly to reproduce. For example, in Drosophila, as you can see up there, many nurse cells have to pour their contents into a single egg. And then, in many species, those eggs get brooded, they get carried, sometimes they get nourished, and then eventually they hatch. And after they've hatched, what do? Sometimes we lactate or otherwise feed these offspring as they're growing and growing. And then even after they look ostensibly finished, we can still go and provision for them, sometimes for a very long time. So new life is always subsidized. And generally, it's the parents who pay for it. But not always. And that's what I'm going to tell you about today. It's something weird and creepy that we discovered in the course of our Human Frontiers grant on comparative ant social physiologies. So my lab studies socially transferred materials, like all of these beautiful social exchanges, or social fluids for short. And so these are components that are made in one body that get transferred to another body where they act. And I'm very happy to talk about them at length. Unfortunately, I don't have that kind of time right now. So I'm going to tell you something that I've come to appreciate more and more as I've studied the evolution and consequences of these socially transferred materials, is that they enable metabolic division of labor. So metabolic division of labor is the partitioning of a metabolic process into elementary metabolic tasks that are performed by separate units within a cooperative entity. To consider that visually, this is metabolism. It's a lot of work. It's the mess of reactions that are required to produce biomass. Now, here's a simplified version of it. Now, each of those steps is costly. We have to encode, transcribe, translate each of these enzymes, and then we have to power them. We have to provide ATP or concentration gradients. Oh my God, so much work. So wouldn't it be nice if, instead of having to do all of that work, someone could just give you an intermediate, and you only had to do about half of it? So this is metabolic division of labor. And so connecting two kinds of metabolic networks like this can allow systems to circumvent trade-offs. So building metabolic division of labor allows the decoupling of resource acquisition and use over time, across space, and across individuals. And this, in turn, allows for the exploitation of vast new niche spaces. And so we can see examples of this in parental care. We also see it in development, in multicellularity, and in symbioses. Okay, now back to ants. So ant colonies work through division of labor. This is what they are absolutely the best at. So usually you have a queen who mates with one male one time, and then throughout her entire life, she continues laying eggs that go fertilizing from that one mating, and that go on to become workers that create this giant colony, and eventually she might produce some new queens for dispersal. Now, the workers also live very short lives. So two things to notice in this. In social insects, usually a lot of reproduction goes with long life. And this is an interesting inversion from solitary organisms. And then the second thing is that in most ants, whether you're a queen or a worker is fixed from birth. But this is actually rather variable. Ants, one of the things that's amazing about them is there are about 15 to 20,000 species out there, and they represent very different types of social life. So what we often think of about ants is that you have a nice queen and a worker like this. Big queen, tiny worker. That's a queen-worker dimorphism, which I've shown over here on the phylogeny of ants, color-coded on the side. And so you can see that in the clade at the top, the poneroid ants, generally the queen-worker dimorphism is very low. They're about 1-to-1. But then in this very large clade of the formicoids, you can see there's a very wide range, up to 300 times larger queens than workers, which is wild. And so In the ants with high queen worker dimorphism, we can really consider them like superorganisms, like germline soma, very beautiful. But in the other ants, it's less superorganism and more of a society where sometimes totipotent individuals can compete to be the one who gets to reproduce, like these beautiful ants who are fighting for reproductive control. So one thing that really struck me when I learned about this incredible diversity was the variable diets and nutrition flows that you see across ants. So many of the high dimorphism ants actually eat sugary foods, and they feed their larvae through direct one-to-one interactions through social regurgitation of something that's sort of like a crop milk, a specialized crop milk, kind of like royal jelly. And on the other side, many of these low dimorphism species, both the adults and the larvae feed directly on insect prey, raw prey. And so it's sort of an equivalent food. And in some cases, those larvae regurgitate back to the adults, so we have another food flow. And because of that, they've been sometimes referred to as a digestive caste. But then, in some weirder and stranger ants, that digestive caste becomes the digested caste through something called non-destructive cannibalism, where the adults, most often the reproductive, feed on the hemolymph of their very own larvae. So, very creepy. And this is not just a one-off thing. It's evolved many times. And for example, in the genus that I'm showing here, which is Platythyrea, this is so consistent that across the genus, all the third instar larvae have this kind of tubercle, like a nipple, on their belly that the adults will drink from. And that makes it a little bit safer for them than an incision, which you find in other Dracula ants. And of course, I'm going to show you a video. So if you watch here, look at the ant on top up there. She's the one who's going to do it. At some point, she decides, oh, no, oh, this is the time. And then she starts hunting around for that tubercle, and she starts drinking. And the other ant is just keeping the larva in place. And you can see that she's doing rather stereotyped movements with her antennae and with her forelegs as she drinks from her larva. Very creepy. So why would you steal from a baby? So when I first learned about this behavior, I thought, this is absolutely wild. And as somebody who's interested in social fluids, I knew it must be interesting. And so my thinking was essentially that it might be some sort of intermediate from a system where there is no strong metabolic division of labor, no strong social fluid sharing, to the modern ants that we know today who rely very heavily on these social fluids and this metabolic division of labor. So could it be some sort of strange intermediate? So that's what the Human Frontiers grant was, and this was definitely one of those stories where I started Googling people to see who could help me do this, with the idea to look at what are the physiological flows across social tissues in ant colonies. And so the idea was to take a few different species from the poneroids, a few from the formicoids, and to use multi-omics, so proteomics, metabolomics, transcriptomics, to trace the sources and sinks and these flows across the colonies, paired with genome-scale metabolic models. And so to do that, I reached out to Bas Zeusink, who does a lot of genome-scale metabolic models, mostly in things like yogurt. So a little bit different. But he's wonderful, and it's been a fantastic adventure. And also Brian Fisher, who is an ant systematist and taxonomist, and has an incredible encyclopedic knowledge of ants around the world and where to find them. So we started this wonderful collaboration. You can see us here in Madagascar, which is where the ants I'm going to tell you about come from. And while I don't get to tell you about the vast multi-omics project and the genome-scale metabolic models yet, I will tell you about this quirky bit of biology. So before behavior, we need to get the ants, right? So these ants live in Karindi Forest in Madagascar, near all those baobabs. And Brian has an amazing team at the Madagascar Biodiversity Center, Gigi and Fidel, who helped then postdoc Andy Brown to go collect these colonies. So they managed, they got them out there in the forest. But then, where's the queen? How do we study this when where's the queen? None of the colonies had queens. Why? So it turns out this ant genus is incredibly diverse in their reproductive strategies. They can do queen and totipotent workers, they can have all sorts of different things, they can reproduce asexually, and sometimes they do all of the above in the same species. Okay, so what were we gonna do? So we brought them back and we did some behavioral tracking. So this is infrared-based imaging, where we have barcode-based recognition of each of the individuals within the colony, and we image them over days and see who's doing what within the colony, who's laying eggs, etc. So getting movement metrics, nearness to brood, nearness to food, and egg laying. And I don't know if they walked around for you. But basically what we see is that there is a core of individuals, so each of the dots is an individual in the PCA, and then you also see each of the individuals in the tree and in the heat map. And you can see that there's a core that hang out near the larvae, near the eggs, and don't really care about the food. And when we look at who's the egg layer in this colony, there she is. So she's right in the center of this egg-associated, larval-associated community, where she should be if she's drinking from larvae. And when we look at other colonies, we see approximately the same thing. These ones are the ones who are who are very close to the eggs, close to the larvae. They don't move very much. They don't get out of the same little region. Except we also sometimes find a sneaky layer. So up here in the orange star, we found 2 layers in the same colony in the same few days, one who was in the right place doing what she should do, and one who just laid an egg in a corner and tucked it into the egg pile. So that tells us already that we're going to find some interesting reproductive dynamics in these colonies. So then which ant drinks and what happens when they do drink? So here Hugo and Nadine set up some experiments. So basically they took each individual from a colony and then gave them either access to one of these late instar larvae or no access. They both had external food. And we did this in 2 ways, one by splitting single colonies and one by giving each colony all access or no access. And then we watched what happened. And what we saw was that when solo adults were given access to these larvae, about 2/3 of them would lay eggs. And when they were not given access, 0 laid eggs. And when we looked at how long it took them to lay eggs, they laid eggs within about 6 days on average. And that's pretty quick to go from being a worker to suddenly being a reproductive. And so given access, 65% lay eggs, typically in less than a week. So that was really exciting, and it suggests that access to larvae is essentially required to reproduce, with implications for how these colonies can disperse. And then we also noticed that when they had access or did not have access, there was also a longevity association. So when the ants who had access to larvae lived longer than controls, the controls are mostly gone by day 10. And so so that presents a question. But the problem is that if you die, then you can't lay an egg. So maybe there's a confounder. So we also looked at amongst the ants who had access to larvae, we also could see this difference in whether they laid eggs or not, in whether they died or not. And then putting those 2 things together, we could look at within access comparison to see that amongst all the ants who had a larvae, the ones that became layers were outliving the ones who did not. So also exciting. But then why and how are they actually living longer under these circumstances? So behaviorally, who are the layers? So in one of the experimental runs of these little dyads of larvae and adults, Hugo was monitoring them several times a day to watch what they were doing. And what he could see is that the individuals who went on to become layers were doing brood care, tubercle feeding, and they were not foraging. So we could really see a big difference that the ones who were laying eggs were the ones who were drinking from the larvae. So they spend their whole day drinking and tending the brood, while the non-layers are the ones foraging. So what is this miracle elixir that seems to give ants both fertility and longevity? Probably wouldn't work for us. So Yuchi Raitsimawang was doing much of the multi-omics of the project, and a little tidbit about this is we can see when we compare the tubercle secretion to the hemolymph of the larva, we can see that it is different, and there are a number of things that are upregulated. And then when we compare that tubercle secretion to the contents of an egg, the eventual egg that a reproductive produces, we can see they're also quite correlated, with the exception of the larval storage proteins, which are much higher in the tubercle fluid. So essentially, the larvae are providing the adults all the materials they need to create an egg, plus a little bit of extra storage proteins that they can use to power the situation. We thought, how is this possibly happening? How is it getting translocated through the body? So we did a bit of backscattered electron SEM with the microscopy facility in Cambridge. And so this is a cross-section of the abdomen of a reproductive. And so you can see the midgut as the big single thing, and then you see 3 developing eggs around. Okay, they're doing what they should do. And when we look at what the tubercle fluid itself looks like, this is what we see. So it's full of lipid droplets, maybe even something like yolk granules. We still didn't see something obviously translocating. So we can see that these structures are still separate. So we're still trying to figure out how this is working, but it might very well work the way it does in many organisms, that these things, lipoproteins, et cetera, get uploaded into the hemolymph and then downloaded into the eggs. So to summarize, Platythyrea, stealing from babies to make babies. So these are queenless ants. We see totipotent workers, and the larvae secrete a fluid that the adults are drinking. The egg layers are usually these brood-embedded individuals. They drink the fluid, but they contest access, and there are sneaky layers. And we see, I think the most exciting is that this access to this tubercle fluid seems to be the thing that's gating reproduction in this species, which is a new form of reproductive division of labor. And it goes to the longer life. So it seems to be enabling a certain type of metabolic division of labor that enables the reproductive to live longer than everybody else. And this secretion seems to carry the ingredients for eggs. So to summarize, reproduction is always subsidized by a division of labor amongst cells, amongst tissues, and here amongst individuals in a very creepy fashion. And then a big, enormous thanks to the Human Frontiers, because this is the kind of project that is kind of wild. And I think it's absolutely fantastic that it's been funded, and thank you for letting me connect to these people. There are a lot of things that we're looking forward to doing next, but one of the things that's been great is that it's led to a BBSRC grant that's completely about this type of question of metabolic cooperation between bodies, and we have a continuing collaboration with the Madagascar Biodiversity Center on other things. So big thanks to the team, and happy to take questions if I have time.